Good morning, and thank you all for joining today's Science, Conservation, and Humanities lecture presented by the New York Botanical Garden. My name is Charles Zimmerman, and it is my pleasure to welcome you from locations across the United States and around the world to today's webinar, Herbaria, Collectively Saving Plant and Fungal Biodiversity. At any time during today's event, please feel welcome to submit your questions by clicking the Q&A button at the bottom of your window, and our hosts will raise these for our speaker to answer at the end. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. Now it is my great pleasure to welcome J.V. Kosoboom, Interim CEO and President of the New York Botanical Garden, who will introduce today's speaker, Dr. Barbara Thiers.
Thank you, Charles. On behalf of NYBG, welcome everyone. It is truly my honor to introduce our speaker and my colleague for more than 2 decades, Dr. Barbara Thiers, Vice President, and Patricia K. Holmgren, Director of the New York Botanical Gardens William and Linda Steer Herbarium, as we celebrate the publication of her remarkable new book, Herbarium, The Quest to Preserve and Classify the World's Plants. In this presentation, Barbara will draw upon her considerable expertise and leadership in the field of biodiversity collection science to share a fascinating history of herbaria and their role in our modern world. Dr. Thiers began her long and distinguished career at NYBG after receiving her doctorate at the University of Massachusetts in 1981. Since the year 2000, she has been responsible for overseeing NYBG's remarkable collection of 7.9 million preserved specimens of algae, bryophytes, fungi, and vascular plants. For decades, Dr. Thiers has been involved in national initiatives to digitize natural history collections in the United States, excuse me, and has been instrumental in setting up the CV Star Virtual Herbarium, a searchable database of all digitized specimens at NYBG. Barbara's numerous leadership roles include managing editor of Index Herbariorum, the definitive guide to the world's approximately 3,400 herbaria, president of the American Society of Plant Taxonomists, past president of the Society for the Preservation of Natural History Collections, board member of the Natural Science Collections Alliance, and member of the External Advisory Board of iDigBio, the National Collections Digitization Hub. When time allows, Dr. Thiers' own research on liverworts brings her on exploratory trips across the globe from Australia to Latin America and the Caribbean. We are enormously grateful for the work Barbara has done to position the New York Botanical Garden Herbarium as one of the foremost in the world. And we are so happy Barbara is here with us to share her expertise on the history and future of herbaria, which has culminated in the impressive publication we celebrate today. Please join me in welcoming Dr. Barbara Thiers.
Well, thank you very much, Charlie and JV. For the past almost 6 centuries, scientists have been documenting the plants and fungi of the world through herbaria. This fairly simple means of preserving biological tissue, basically drying and gluing to sheets, was a key innovation in the study of plants and fungi that made it possible for this subdiscipline to grow into an independent scientific endeavor. The herbarium made it possible for scientists to characterize those plants that grow in faraway places and to understand their diversity in a global scale. In my talk today, I want to tell you a few stories that I especially like from the history of herbaria. I'll also talk a bit about how herbaria contribute to understanding the environmental challenges we face today. Most of the content I'll present here is from my recent book on herbaria, published by Timber Press last month. I've been around herbaria all my life. My father was founder and, for nearly 40 years, curator of the herbarium at San Francisco State University. In addition to an unrelentingly heavy teaching load, he poured his considerable curiosity and energy into documenting the fungi primarily, but also the bryophytes and lichens of California. My childhood weekends were spent either on field trips or in the herbarium, where I could earn some pocket money by helping my dad with his specimens. Honestly, the money was far more motivating to me than any love of herbarium work at that age. At one point, I had a specific fundraising objective in mind. In the worst way, I wanted a pair of white go-go boots, which were all the rage in the late 1960s, and which my mother refused to buy for me. Once I had enough money to buy my own boots and was in the thralls of teenage rebellion, I wanted nothing more to do with the herbarium, or my parents for that matter. But in the middle of my college years, I found my way back. I discovered that I too wanted to participate in the great adventure of documenting plant and fungal life, and to join my dad and his passionate, often eccentric band of students and colleagues who derive such pleasure from this work. I arrived in the New York Botanical Garden in 1981, as JV mentioned, to start a one-year postdoctoral fellowship in the herbarium. My project was to organize and index the collections of the William Mitten Herbarium. Mitten was a 19th century English apothecary and self-taught botanist who obtained specimens from essentially all the famous European exploring expeditions of the 19th and sometimes even 18th centuries. The work was thrilling but frustrating because often these historic specimens lacked adequate documentation. A scribble and written in terrible handwriting, as exemplified by the images of actual specimen labels shown here, might be all that accompanied a historic specimen. From that experience, frustrating as it sometimes was, I developed an enduring interest in botanical exploration and documentation of historical collections. I managed to stay on at the garden past that postdoctoral year and worked my way up through the administration of the garden's William and Linda Steer Herbarium. Among the many things I have loved about working in a herbarium has been the opportunity to visit and get to know the staff of herbaria all over the world. These institutions differ in language and culture, but all are committed to preserving and sharing their wealth of plant and fungal specimens. At a time when we seem to be bombarded daily by negative aspects of human nature, herbaria highlight one of our better human impulses to save things, not just for ourselves, but also for generations to come. It is true that many times plant and fungal collections have been fueled by imperialism. The impulse to kill these organisms just to store them in cabinets may seem to derive from some base hoarding instinct. However, investing time and resources to care for these irreplaceable representatives of past life and to share them freely with anyone wanting to study them is both generous and wise, because herbarium specimens constitute a unique source of data that help us understand how the world's vegetation has changed over time and to predict how it will change in the future. Evidence suggests that this man, Luca Ghini, created the first herbarium. Ghini was from the region around Bologna. He lived at a time when the Renaissance was starting to blossom in northern Italy, and forward-looking universities in the region encouraged independent thought, and new ideas and goods came from travelers passing en route from northern Europe to the Mediterranean and the Middle East. After graduating from the University of Bologna with a medical degree, Ghini was hired there as a professor. He offered what is generally believed to be the first botany course offered in the medical school with a focus on medicinal plants. Ghini's course was very popular with students and within a few years was elevated to a core element of the curriculum. Instead of relying purely on classical texts such as Dioscorides' Materia Medica, Ghini encouraged his students to make independent observations of the plants that he collected for their study. He maintained these reference specimens as pressed and dried plants glued to bound pages of a book. Later, he created the first botanical garden in Pisa when he moved on to teach at the university there. Like his herbarium, the aim of the garden was also to aid in the teaching of botany. Botanists came from all over Europe to visit Ghini, and they adopted the herbarium technique to aid in their plant study. Ghini's herbarium has not survived, but those of some of his students still exist, and some examples of their specimens are shown here. Leonard Rauwolf, a German botanist who was among the first Europeans to collect in Turkey and the Middle East, created the most beautiful of the early herbarium volumes. For reasons that aren't entirely clear, he bordered the pages of his herbarium sheets with a type of wallpaper that was designed to cover wooden beams in a building. As mentioned earlier, herbaria contain not just vascular plants, but also algae, bryophytes, that is, mosses, liverworts, and hornworts, and fungi, including lichens. Sometimes collections of fungi only are referred to as fungaria. Most of the 16th century botanists largely ignored these organisms because it wasn't until the microscope was invented that their life histories could be understood. Also, both mushrooms and some algae may decay quickly after collection and are difficult to preserve. However, a few bryophytes and lichens and the occasional alga can be found among the pages of the earliest herbaria. In all of these groups, the features that are key for understanding their life histories are too small to view with the human eye alone, which inhibited an understanding of their relationships to other organisms. In fact, because of the inability to see the reproductive structures in these organisms, they were often referred to as cryptogams, meaning hidden gametes or sex organs. The earliest herbaria that focused on these so-called cryptogams were those of Pier Antonio Micheli and Johann Dallenius. Micheli was the first to describe reproduction in fungi, and thus he is often considered the founder of mycology, the study of fungi, and is also known for his pioneering studies of bryophytes and lichens. His herbarium, which also includes that of his students, is maintained at the University of Florence, and it contains about 19,000 specimens. Dallenius, a German botanist hired by Oxford University, was very much influenced by Micheli. His herbarium at Oxford consists of about 900 specimens on 176 sheets. At the time that Guinea was inventing the herbarium, other world cultures were building upon centuries of plant knowledge that surpassed that of Europe in volume, longevity, and continuity. Chinese scholars produced a new and original work on pharmaceutical botany at least every century, beginning in 300 BCE. The unbroken advance in plant knowledge over time in China led to a consistently high level of accurate description and illustration, and a standard method for naming plants. The impetus for plant knowledge in the Islamic world had a spiritual as well as a practical basis, because descriptions of paradise in the Quran included the names of the plants that grow there, and because the plants are invoked frequently in stories of the Creator's power. One of the earliest manuscripts for which a part still exists is the Kitab al-Nabat, or Book of Plants, that describes some 600 species of plants. Many publications of a similar nature followed this one, documenting the plants and their uses throughout the far-flung Islamic world. The Aztec king Montezuma maintained botanical and zoological gardens that were said to be far more advanced than those in Europe. Aztec libraries contained thousands of manuscripts that sadly were burned in the European conquest of their territories, destroying all records of pre-colonial scholarship. Most of what we do know about Aztec plant knowledge today comes from the work often referred to as the Bedenius manuscript, written by two Aztecs who assimilated into colonial culture. Interestingly, none of these cultures developed the tradition of herbaria. In the seasonal European climate, many plants cannot easily be observed all year long. However, in other parts of the world with a tradition of written scholarship where the climate is more moderate, plants could be observed for a longer period of the year. So there was less need to use preserved plant material to study plants. Also, none of these cultures but European had a prolonged period of intellectual stagnation like the Middle Ages, which interrupted the tradition of recording natural history information that had been established by classical authors. When science reemerged in the Renaissance, European scientists lacked a knowledge base that had been accruing century by century, forcing them to start afresh to build this knowledge. The lack of precedent and an intellectual climate open to new ideas are thought to have spawned inventions such as the telescope and the microscope, and perhaps herbaria and natural history collections in general fall into that category as well. Beginning in the 1600s, European countries invested heavily in sea and land-based exploration, the goal of which was to establish colonial outposts where they could exploit local resources for products for trade and to secure safe and dependable routes to transport those goods back home. Many of the products they were keen to obtain were plants or plant-derived, most notably spices, but also horticultural plants like tulips, which sold for astronomical prices in the early 1600s. Although the botanical objectives of a government-sponsored exploring expedition usually focused on a few species of commerce, by the beginning of the 18th century, the idea emerged that at least some expeditions should include naturalists to document the native vegetation and animals of the foreign lands visited. Such trips resulted in the collection of living plants as well as those preserved as herbarium specimens. Only about 12,000 of the estimated 400,000 known species of plants occur naturally in Europe, so almost every trip brought home plants that no botanist there had ever seen before. As a result of this influx of exotic flora, understanding the breadth of the world's plant diversity became a central focus of botanical study. Those charged with plant collection on an expedition would make sorties from the ship or land-based camp looking for plants suitable for collection, which would generally be those displaying as many of the features needed for identification as possible, namely leaves, buds, flowers, and fruits. Portions of such plants would be placed in a cloth sack or a vasculum, which is a metal case in the shape of a somewhat flattened cylinder that opens along the long side and has a handle or strap for ease of carrying. Pressing of the specimen takes place as soon as possible after collection. This procedure begins with the laying out of the plant between a sheet of paper in a way that keeps all key parts visible and not overlapping one another as much as possible. The paper can be any absorbent type, but newspaper is often used today. Then the plants and their papers are stacked in a press, which consists of two rectangles of either solid boards or cross-tapped strips of lathe that are held together with straps or screws. Once pressed and dried, the specimen is glued to a stiff sheet of paper, positioned in a way that's both attractive and shows the important features of the plant. Also glued to the sheet is a label on which is given information such as the scientific name and when, where, and by whom it was collected. Older specimens, as I showed you previously, sometimes lack the full complement of information that's found on the label shown here. The first European explorer charged specifically with the collection of plant specimens for scientific purposes was a sort of unlikely figure. William Dampier, a working-class Englishman who started his life at sea shortly after completing his primary education. As a young man, he worked on ships that harvested logwood, a tree in the legume family, along the shores of the Caribbean Sea. The wood was used back home for dyeing wool and fabric. Later, Dampier made his way around the world as a privateer, which is a sort of government-sanctioned pirate. A privateer had permission to attack foreign ships during wartime and take the ship and its contents as a prize. And proceeds were then divided among the sponsors, the ship owners, the captain, and the crew. Dampier kept a very lucid and detailed written account of his travels on this trip, not only of his plundering adventures, but also the sea currents, landforms, people, and plants and animals he encountered. He went to great lengths to maintain his journal, placing the rolls of parchment within hollow bamboo stems, the ends of which he sealed with wax. When Dampier returned to England from this trip, he was introduced to a new phenomenon that had developed in his absence, the coffee house. An interesting feature of the English coffee house of this time was that these were one of the few places where the social classes mixed. Dampier frequented Jonathan's, a coffee house in Change Alley in central London, whose location is still occupied by a cafe. There, he rubbed shoulders with powerful people who were fascinated by his stories, which prompted him to write a book about his travels entitled A New Voyage Around the World. It was hugely popular and it went through 3 printings and was translated into Dutch, French, and German. Support from the friends he made at the coffee house and the success of his book led the Royal Navy to sponsor a second expedition for Dampier. He was to survey Terra Australis, which included Australia, but was thought to be a much larger landmass. The English Navy was especially interested in having him explore the eastern coast of this continent, which many thought extended all the way to the South Pole. Although enthusiasm was high for the voyage, the ship assigned to it was a little disappointing. The HMS Roebuck was a 9-year-old fire ship, one that, being near the end of its useful life, was slated to be used as a weapon. In the height of a close naval battle, the ship would be filled with combustible contents, sent into the enemy fleet, and then ignited. Dampier's expedition departed in 1699. His route took him first to Brazil, as did most expeditions to the Pacific, but instead of sailing around Cape Horn, Dampier headed east from there, sailing farther south than most other ships, taking advantage of the strong winds of the so-called Roaring Forties. About 4 months after leaving Brazil, Dampier and his crew reached the western coast of Australia, low on supplies of food and water, but without any loss of crew to scurvy or other mishaps. The first Australian landfall was on Dirk Hartog Island in Shark Bay, approximately here. Dampier collected about 20 flowering plant specimens and one marine alga there, which he pressed between the pages of a book. Although he complained that some specimens were spoiled, those that survived were very well preserved and perfectly adequate for scientific study. At another Australian landfall, he discovered his most spectacular flower, the plant now known as Sturt's desert pea. From Australia, the expedition sailed northwest to New Guinea. As the trip progressed, however, Dampier became increasingly concerned about the state of his ship, whose hull was badly damaged by shipworms. He decided not to continue on to the eastern coast of Australia, but instead to begin the trip home. The Roebuck made it most of the way back to England, but sank off the coast of Ascension Island. They were near enough to land when the ship sank that all the sailors survived the wreck, but many of Dampier's specimens were lost. Several months later, the crew was rescued by a passing Royal Navy ship, which returned them to England. Surviving botanical specimens included plants from northwestern Australia and a moss from New Guinea, but not much more. These were deposited in the Herbarium of Oxford University, where they remain today. About 17 species based on his collections were described by botanists of that day, but interestingly, Sturt's desert pea would not be described for another century. Dampier wrote a book about his second trip as well, but it was not as popular as the first. He was dismissed from the Navy because he lost the Roebuck and because of an issue with his second in command, who he had taken off the ship because he feared he was plotting a mutiny. Although he made many more sea voyages as a privateer, Dampier didn't collect any more specimens or write any more books. I was interested to learn that Dampier's books contained the first written usages of more than 1,000 words, according to the Oxford English Dictionary, including such commonly used words as barbecue, avocado, chopsticks, and tortillas. Finding Terra Australis was also the goal of the Bougainville Expedition of 1766 to 1769, sponsored by the French government. Philibert de Commerson was appointed as naturalist of the expedition. He was a landowner and physician whose passion was plants, which he'd been collecting and growing since he was a boy. Probably in the pursuit of collecting, Commerson met Jean Barré, and they became lovers. Barré was an herb woman who earned a living collecting medicinal plants and selling them to physicians and apothecaries. Commerson's terms of service with the Bougainville expedition stipulated that he could bring an assistant of his choosing, and he chose Barré. However, a royal ordinance forbade women from sailing on French naval ships, and sailors had strong superstitions that a woman on shipboard meant bad luck for the voyage. So, shortly before departure, Commerson came aboard and settled into his quarters. Barré appeared a little later, impersonating a young man looking for work on the expedition. Commerson pretended to interview her for the job as his assistant, and after satisfying the captain that he was a good fit for the job, she came aboard and moved into Commerson's quarters. It was a good thing for the scientific mission of the expedition that they pulled off this subterfuge, because Barré was the one who did most of the collecting. Commerson was never in good health on the voyage. The first place they collected extensively was Brazil, after which the voyage headed south around Cape Horn to the Pacific. And after many months at sea, they made landfall in Tahiti. It was about this time that Barré's identity was discovered. Bougainville chose not to punish her, but there is strong evidence that she was subsequently raped and mostly kept to her own quarters after that. When the expedition arrived in Mauritius, a man named Pierre Poivre, who was the founder of the botanical garden there, requested that Commerson stay to help him carry out botanical explorations. Fun fact, Poivre is probably the protagonist of the tongue twister, Peter Piper picked a peck of pickled peppers. Poivre translates to pepper in English, and peppercorns were a major export from Mauritius, and they were usually packed in brine for transport. It was convenient for Bougainville to grant Poivre's request so that his triumphant return to France would be untarnished by the revelation of Jean Barré's true identity. Since Barré was noticeably pregnant by the time they arrived in Mauritius, such a discovery was a foregone conclusion. So Commerson and Barré and all of their collections stayed behind when the expedition left for home. Barré continued to conduct fieldwork with Commerson after giving birth and relinquishing her child for adoption. Commerson's health continued to decline, and 4 years later, he died in Mauritius, leaving Barré without an income or a place to live. Ever resourceful, she opened a tavern in the capital city, and the following year she married a former French army sergeant. They subsequently returned to France, and Beret thereby became the first known woman to circumnavigate the earth. Beret's homecoming was not officially noted, but she was granted a pension, and with these funds, plus a bequeath from Commerson, Beret and her husband lived comfortably in her husband's home village of Saint-Olay near Bordeaux until her death in 1807 at 67 years old. I went to France in October of 2019 to see her grave, and I was delighted to find that it is well documented and maintained, and clearly fairly often visited, judging by the number of offerings left there. After Commerson's death, the specimens were sent back to France. The shipment contained some 30,000 specimens representing 5,000 different species, of which 3,000 were considered new to science. This was by far the largest collection of natural history specimens ever made on an expedition. And as I mentioned earlier, most of these were made by Jean Beret, though none of the specimens attribute collection to her. Their specimens are now stored in the National Herbarium of Paris, and they've been digitized and can be viewed online. The most famous plant from the expedition is Bougainville spectabilis, the extremely popular ornamental vine that is now grown all over the world in warm climates. Beret's botanical contributions were finally commemorated many years later through the description of the South American species Solanum barretti, which was described by Eric Tepe of the University of Cincinnati and co-authored by colleagues that included Beret's biographer. The first government exploration in the United States that resulted in herbarium specimens was that of Lewis and Clark. The expedition had the goal of exploring the newly acquired Louisiana Territory and opened the door to the westward expansion of the country and the displacement of native people from their land. Meriwether Lewis and William Clark were trained to make botanical specimens by Philadelphia botanist Benjamin Smith Barton, who was also supposed to be responsible for the identification of the specimens after they were collected. Barton, however, turned that responsibility over to a German botanist named Friedrich Persch, who came to Philadelphia for a while in the early 1800s and was mentored in his botanical studies by Barton. Persch did some preliminary work on the specimens while in Philadelphia and turned many of these over to the American Philosophical Society for their herbarium. Then he returned to Europe, taking the remaining Lewis and Clark specimens with him, apparently without knowledge or permission. In England, he continued his botanical work with the support of Aylmer Lambert, a wealthy botanical hobbyist with an important private herbarium. Under Lambert's patronage, Persch published his two-volume work on the North American flora in 1817, the most comprehensive treatment of American plants to date. In this work, he described 71 new plants based on Lewis and Clark's collections, including the genera Lewisia and Clarkia. Persch gave the Lewis and Clark specimens still in his possession to Lambert. And when Lambert died a few years later, his herbarium was put up for sale at auction. Half of the American collections in Lambert's herbarium, including Lewis and Clark's, were purchased by a young American botanist named Edward Tuckerman, who was making a European tour before settling down to a teaching position at Amherst College. A specialist in lichens, Tuckerman was not interested in keeping the flowering plant specimens, and he returned these, including the 47 Lewis and Clark specimens, to the Academy of Natural Sciences in Philadelphia in 1856. For 40 years, the Lewis and Clark specimens sent by Tuckerman were thought to be the only ones that existed. All the others were thought to have been lost until 1896, when the rest were discovered in storage in the American Philosophical Society attic. These specimens were still bundled in their original pressing papers, and some, sadly, had been eaten by beetles. The Lewis and Clark story highlights a theme that runs throughout the history of herbaria. A huge amount of effort goes into the collection of the specimens, but not nearly as much into the actual study and care for the collections after they're made. To make sure that specimens are properly managed after a collection requires expertise and infrastructures, which are often lacking in the early years of a new nation. Despite the lack of interest in the Lewis and Clark specimens in the years immediately after their collection, today these specimens are preserved in a manner befitting their national importance. There are in total 222 specimens, all of which have been digitized and stored in a climate-controlled room designed specifically for them. After Lewis and Clark, the US government sent many additional expeditions to explore the Western US. The botanical collections from these were handled much more efficiently than with Lewis and Clark's. Most went to John Torrey and later Asa Gray for identification. Torrey was a New York City native who became the first internationally recognized authority on American plants. He planned to write a flora of the entire United States, but he never completed it. However, he described many new species and amassed an herbarium of an estimated 50 to 80,000 specimens, most of which he mounted himself and annotated with observations and comparisons of species. In 1860, when the collection outgrew the available space in his living quarters, Torrey donated his herbarium and library to Columbia College, where it remained after his death and eventually formed the nucleus of the New York Botanical Garden Herbarium. Asa Gray, as a student of Torrey's, took over his role as the leading American botanist of the latter half of the 19th century. His herbarium is the centerpiece of the Harvard University Herbaria, which today, with an estimated 5 million specimens, is the largest university-based herbarium in the world. Although Torrey and Gray were the dominant forces in documenting American plant diversity in the 19th century, as the population moved westward, centers for plant study developed across the country. After the gold rush created the city of San Francisco from the quiet mission town of Yerba Buena, centers for botanical study developed on the West Coast at both the California Academy of Sciences and the University of California at Berkeley. We'll focus on the Academy, although the history of botany at Berkeley is equally fascinating. The California Academy of Sciences was founded in 1853 with the mission of documenting the plants, animals, and geology of the state. The Academy had an interesting clause in its charter, indicating that the institution, quote, highly approved the aid of females in every department and earnestly invited their cooperation, end quote. It thereby became one of the first institutions in the country and possibly the world to recognize and encourage women scientists, a decision that paid off for the institution in spades due to the efforts of two highly accomplished and energetic women that it employed. Kate Brandegee, who trained as a physician at UC Berkeley, was hired as a curator at the Academy's herbarium. She and her husband, Townsend, collected extensively throughout the state and also in Baja California. Kate eventually gave up her job as curator to focus solely on plant collecting, and to replace her, she hired Alice Eastwood, a talented young botanist from Colorado. Eastwood was born in Toronto, but moved to Denver as a young teen. Her early love of nature, especially plants, grew during long hikes in the Rockies. She was a talented student, graduating from high school as class valedictorian. However, her family's precarious financial situation precluded the possibility of higher education for her. Instead, she taught high school, but continued to spend as much time as possible exploring the plants of the Rocky Mountains on her own, and she developed a local reputation for her trail expertise and plant knowledge. When Alfred Russel Wallace visited Denver in 1887, it was the 28-year-old Eastwood who took the famous tropical explorer, who was then in his 60s, on a 3-day hike to the 14,000-foot summit of Gray's Peak, introducing him to Colorado's diverse and beautiful alpine flora. A lucrative real estate sale allowed Eastwood eventually to quit her teaching job and travel, and that's when she met the Brandegees. Eastwood's energy and enthusiasm for the study of plants and her dedication to the Academy's herbarium gained worldwide appreciation through her heroic efforts to save the herbarium during the earthquake and fire in San Francisco on the morning of 18 April 1906. Eastwood felt the tremors that morning, but since it was not strong in her neighborhood, she went to work that morning with no particular anxiety. However, when she reached the Academy, she saw that the building was damaged, and worse, the surrounding buildings were on fire. The marble staircase leading to the 6th floor of the building where the herbarium housed had collapsed, but Eastwood managed to climb up the banister, which was still intact. From the herbarium windows, she could see that the fire had reached the adjacent building and that she had only minutes to save what she could. She chose to save the approximately 1,500 type specimens, the most valuable part of any collection, and her Zeiss lens. She bundled the specimens in a large piece of fabric and lowered them by rope to the ground. By the time she got back downstairs, police and firefighters were desperately trying to clear the area as the fire closed in. Eastwood secured the specimens on a horse-drawn cart and took them to safety. In a letter to Science magazine, published some months later, she wrote, "It is a great loss to the scientific world and an irreplaceable loss to California. My own destroyed work I do not lament, for it was a joy to me while I did it, and I can have the same joy in starting again." When the Academy reopened its doors in 1916 at the new quarters in Golden Gate Park, Eastwood went right to work rebuilding the collection. Under her care, the new Academy Herbarium grew from the nucleus of saved specimens to reach a total of 300,000 by her 80th birthday. Today, there are approximately 3,400 herbaria in 180 countries around the world, and collectively they hold approximately 400 million specimens. Herbaria still serve their original function to document the occurrence of plants and fungi and to provide a reference for their identification and characterization. However, recent technological advances have allowed us to broaden the uses of herbarium specimens. Understanding the impact of global change on Earth's organisms and ecosystems has become a central focus of biological research today. But documenting such change scientifically is tricky because it requires measurements and observations before and after the change event. Herbarium specimens provide one of the very few tangible sources of data about how plants and fungi lived before industrialization and through each successive period of technological advance since that time. Because plants are rooted in the ground, they are particularly exposed to environmental conditions in the place where they grow. As you know, heavy metals such as cadmium, lead, and mercury have adverse effects on animal life, even in tiny quantities. These elements can reach harmful concentration in our soil and drinking water as a result of unregulated agriculture and industrial processes, as well as inadequate waste disposal methods. Generally less sensitive to these chemicals in animals, plants and fungi absorb heavy metal in the water they take in through their roots or hyphae and are retained in their tissues in herbarium specimens. Therefore, we can use specimens to determine the concentration of heavy metals to get a historical perspective on heavy metal pollution in the past. A variety of studies have shown that heavy metal concentrations in herbarium specimens from industrialized regions are highest from the times when manufacturing had few pollution control measures, especially in the late 19th century in Europe and North America. A study at Brown University, published in 2017, of herbaceous plants in Rhode Island, collected between 1846 and 2015, demonstrated that lead concentration in plant specimens decreased during this period as more effective emission controls were applied to manufacturing plants in the area. Interestingly, lead levels were also high in plants from the offshore Block Island, 13 miles from the mainland, where there was no significant industry, indicating the breadth of area that this pollution affected. In the 1990s, scientists discovered that gene sequences could be recovered from the tissue of herbarium specimens, and since that time, the process has been refined to require smaller amounts of tissue, and the success rate has increased even for very old specimens. Until recently, most of the DNA-based research using herbarium specimens contributed to delimiting species and constructing phylogenies, which is a fancy way of saying a family tree, which can be used to understand how organisms are related to one another. New and more powerful tools for sequencing DNA, known as next-generation sequencing, now make it possible to sequence all the genes in an organism, or its genome, and such techniques are now being applied to DNA recovered from herbarium specimens. Surveying the differences in genome structure among plants preserved as herbarium specimens allows researchers to understand which genes control traits such as plant height, leaf size, color, flowering time, and how variation in the expression of these traits have developed in different locations and in different lineages of organisms. Current projects using herbarium specimens in genomic studies include those looking for genes related to the development of nitrogen fixation in plants, drought tolerance, and also the traits that make certain species invasive. Specimen digitization is the process of capturing images of specimens, transcribing their label data into a database, and then sharing the results through an online data portal. Today, there are about 80 million digitized specimens from the World Herbaria shared through several different data portals. In the US, most of the digitization work has been done in the past decade, thanks to a special funding program of the National Science Foundation. Digitization has opened up many new uses for herbarium specimens. For example, this map plots the location of herbarium specimens from Brazil held by New York Botanical Garden Herbarium, overlaying map layers showing the distribution of Amazonian forests and the location of fires in the summer of 2019, allowing for a visualization of what plant biodiversity may have been lost through that environmental catastrophe. When making herbarium specimens of plants, collectors are particularly concerned with documenting structures that contain diagnostic features needed to distinguish one from another. Among these features are leaf and flower buds, open flowers, and fruits. These structures also indicate stages of the plant's phenology, or seasonal life events. And thus, in addition to using specimens to identify plants, we can also use them to deduce the timing of their annual growth and reproductive cycles. Because we know when and where each specimen was collected, we can compare phenological change over time and space. With the volume of digitized specimen images now available, it's possible to score large numbers of specimens for their phenological state. Why would we do this? Well, shifts in phenology are one of the most commonly predicted effects of global change and are a cause for concern because such shifts could lead to a mismatch between plants and other organisms with which plants are interdependent, such as pollinators. Studies using large sets of digitized herbarium specimens have indeed confirmed that these predicted mismatches are happening. Estimates suggest that up to 50% of the still undescribed species of plants may actually already be represented in herbaria, but are either misidentified or are in the unidentified backlog that almost every herbarium holds. Ours, the New York Botanical Garden, is particularly large. There is great interest in the moment in applying machine learning techniques such as computer vision and data mining to digitized herbarium specimens. This work is at an early stage of development, but holds great promise as a technology that can build on digitized specimens to broaden the use of herbarium specimens even further. Preliminary attempts to apply machine learning to plant identification from herbarium specimens have shown that algorithms can match images of specimens of unknown identity to a reference set of identified herbarium specimen images with an accuracy rate of between 85 and 90%. And further study of the algorithms and refinements of the process will likely make the rate of correct matching even higher. If we are able to combine machine learning based on image recognition with mining of descriptive data from published literature and gene sequence data, we will have a very powerful tool for plant identification and maybe even one day clear out our specimen backlog. Because of all the digitization and expansion of research uses, the past decade has been a golden age for herbaria. But we face significant challenges at the moment, mostly stemming from the insecurity of funding. Herbarium budgets rarely rise in good times, but they almost certainly fall when economic times are bad. And based on surveys of collections that we've carried out during this pandemic time, there's reason to be concerned about the future of some herbaria and other natural history collections in this country and, of course, around the world. There's no magic answer to how to keep funding, but remaining relevant is certainly key to retaining institutional funding base sufficient to cover the maintenance and growth of a herbarium. The collection must be relevant to the institution that houses it and to broader national and international scientific and educational goals. Last year, a major report sponsored by the UN was published on biodiversity. The report made the argument that increasing public awareness of the importance of biodiversity is as critical to protecting it as the research and conservation efforts. Herbaria have a role to play in increasing public knowledge and appreciation of biodiversity, as well as serving the scientific enterprise. Most herbaria hold periodic events that allow the public in for tours and demonstrations of collecting and mounting and viewing old specimens, although such activities have, of course, mostly been suspended in the past year. At the New York Botanical Garden, we have an herbarium open houses every year. We put out a lot of specimens, each with a short accompanying story, usually around a theme. Our Plants in Love theme for a Valentine's Day open house a few years ago was my favorite. We get plenty of staff to stand by the counters and engage with people who walk by. In addition to specimens and stories, we offer hands-on activities like paper flower making, and we create Instagram opportunities as well. We are very much looking forward to our next open house, though we don't yet know when that will be. The event will be publicized well in advance on our website and social media, so we hope you can come if you can. For those who don't live close enough to attend an open house, you can visit the Handlens, our online vehicle for sharing stories about our specimens. A few such stories are highlighted here. Our hope is that by demonstrating the wide range of people who have contributed to our field, we will broaden the diversity of people who want to participate in this endeavor today. And increasing that participation is critical because despite the centuries of data that we've accumulated to help us understand and protect plant fungal biodiversity, we haven't made nearly enough progress in this work. And incorporating a broader range of voices and perspectives is probably our only hope of doing so before it's too late. At the garden and at essentially all herbaria, volunteers are always welcome, both in person and online. We'll put a link in the chat to a web page that gives more opportunities about information about the opportunities at the garden, but you can check your local herbarium for other opportunities if you live outside the New York area. I'll leave you with one final story that combines the theme of missed opportunities for including diversity and knowledge derived from herbarium specimens, but has a happy ending. Both the illustrations on this slide are of the same plant, Mutissia clematis of the Asteraceae or daisy family, named for the Spanish physician and botanist Jose Celestino Mutis. In the late 18th and early 19th century, Mutis devoted more than 35 years to studying the flora of what is now Colombia, South America, and to improving the lives of people in his adopted homeland. He formed a botanical institute, Casa de Botanica, that employed a large number of local people to collect specimens and then to describe and illustrate them. Some of the people who worked in his casa later became leaders of the Colombian Revolution. The illustrators, including Salvador Risso, whose work is shown here, used pigments from local plants and developed an accurate but also lush and compelling style of illustration that contrasts rather sharply with the image on the right, which represents the more conventional European style of illustration. Some 7,500 paintings like this one were produced by Casa de Botanica, and they, along with the excellent specimens they were based on, might have had a strong influence on 19th century botany. Except shortly after Mutis died, they were confiscated by the Spanish military and taken back to Spain. The specimens and illustrations were hidden away and forgotten about for more than a century, greatly impeding the study of the megadiverse region from which they originated. Once rediscovered, the specimens and images were restored, and today they are beautifully conserved at the Jardín Botánico de Madrid, and anyone can view them online through their website. The rediscovery of these specimens led to a long-standing collaboration between Spain and Colombia to document the flora of that country. I want to end by thanking the New York Botanical Garden for the support of its William and Linda Steer Herbarium, which has been providing critical data for research and education for more than a century and a quarter, and which has given me so many wonderful opportunities in the past 39 years. I also want to thank you for listening. And if this is your first exposure to the wonderful world of herbaria, I hope it won't be your last. Thanks.
Thank you, Barbara. That was so fantastic. We've had so many great comments in the chat as you've been speaking. Thank you. Wow. So this is time now for the Q&A. Audience members, if you haven't already, please go ahead and add your questions by clicking the Q&A button at the bottom. And you're also invited to upvote the questions others have already submitted. So we have some great questions already to get us started. One that came up and was upvoted quite a lot, Barbara. To what extent is indigenous knowledge also recorded in herbarium specimens, both in the past and at present?
So the most frequent time when indigenous knowledge is preserved is when the collection is of a plant that indigenous people use for a particular purpose. So if the plant was used for medicine, for spiritual, or for food, it's often indicated. Um, Standards for doing this have changed a lot. The early collections, it might just be a rather vague note. But more recently, there actually is sort of a standard vocabulary and a standard structure for how you record this information. And so people who are making ethnobotanical collections are very rigorous in how they describe that. There are suggestions for how you record the native language, that it's, that how people say it and exactly how it's used. There are questions about, how you can use this information, and different cultural groups feel differently. There are some who are very eager to have the world know about how they traditionally have used specimens, and there are other people who feel more protective of that information. So today, people are very careful to follow whatever the conventions are that are needed. However, there is I'm sure there are many, many cases when indigenous people contributed important information to the collector about where to collect, about what the plant was or its purposes, that are completely lost.
And as a quick follow-up, do you know whether any herbaria are actually managed by indigenous communities?
Yes, I can think of some in the South Pacific, particularly, where my colleague Michael Balick has worked for years and has been very influential in helping to establish those herbaria, places where people felt that they really should preserve this material as their culture changes, as they saw their flora changing. And so, yes, there are definitely herbaria that are managed by indigenous people. Actually, there are two Native American herbaria in the United States, one in Idaho, and the Navajo Nation also maintains its own herbarium in Arizona.
Thank you. Um, we've had questions about um, the collections worldwide. Do you think we should centralize or decentralize our collections? Is it your take on the existence of large versus small herbaria? What is
Yeah.
their relative importance?
So they have very different importances. First of all, it's important to note one thing about plants. This is a major difference from collections of fish or vertebrates, or mammals, that most people collect in duplicates. So very rarely does someone make just one sheet of a plant they collect. They might make up to 20. It depends on how abundant the plant material is. And the typical convention, if you're collecting in another country, especially, you leave half the duplicates in that country. You bring back to your institution, you bring one to keep, and then you bring one to often to send to someone who for identification, if you don't already know what it is. And if there are duplicates left over from that, then you trade them for specimens from other herbaria. So the beautiful thing about herbarium is that many specimens are duplicated. It's both a blessing and a curse, because actually duplicates end up causing many problems in databases and other places. But so we don't have to completely decide, in the case of herbaria, whether or not they should be centralized or distributed. They can be both to a certain extent. But there are very distinct differences and importances to large and small herbaria. A large herbarium will give you a broad perspective geographically and temporally of the plants of a region. However, a local herbarium may have a very intense set of collections made over of a limited area, but over a long period of time. Many universities, for example, hold collections that are made every year by classes, classes where students went out and collected. And that can be extremely important in looking for fine change over time. So the small herbaria can be extremely important for understanding much finer grained kinds of change than a large herbarium is likely to be.
Thank you, Barbara. And a lot of folks were actually very curious about the Lewis and Clark specimens and the color charts on those specimens. Can you briefly tell
Yeah.
us a little bit more about those?
So the almost all specimens that are digitized have those little color charts on them. And it's really to calibrate the cameras and the use of the images later. Now the plants are, don't look anything like what they looked like when they were alive. They mostly fade. So it isn't so much that you are concerned about the color in terms of identifying the plants. However, you do want to make sure that that the images are not being distorted when they're captured, and you do want to be able to set your computer screen to view the images at the optimal. So when we started this endeavor, we started all using these little color charts on all of our specimens, and you see them essentially on all of them. And it's really just a standard so that the images can be adjusted to be the right, the right quality.
Thank you for that. A lot of folks were also interested, I know we have some history geeks here, about Am Bonpland and Alexander von Humboldt,
Mhm.
their plant collections from South America. They collected more plants than known to Europeans at that point in time in the late 1700s. So what's your take on those expeditions?
I mean, they're extremely important. My understanding is that Bonpland was more responsible for the collections when they were brought back than Humboldt. Humboldt wasn't really an expert in plant identification, and Bonpland definitely was. Um, because that expedition has been so well studied, I did not make it a focus of my study. However, there's wonderful stuff written about them, and their scientific work was breathtaking. They spent some time in Colombia with Mutis, but I sense that there was some delay in it's a story that's repeated over and over again. They made all these great collections, but it took a really long time for them to be distributed. The first set of those would be in Paris, and they would all be digitized and online, but they are very widely distributed. We have quite a few, don't we, Charlie? We have quite a few Humboldt bone plant specimens at the New York Botanical Garden. So yeah, they're extremely important. And of course, that's just one of many things that those expeditions were known for. It's one of the first real studies of tropical plant ecology and biogeography.
There's a lot of stories to tell and just never enough time, right?
Yeah, that's for sure.
We have a comment. Hello, I'm a master's degree graduate in botany from, sorry, I'll mispronounce this, Tribhuvan University, Nepal.
Hmm.
Which online portal is updated and accepted internationally worldwide for binomial names of plant species and mushrooms? Is there one that you follow? She's most familiar with the Catalog of Life.
Right. So just to be clear, she's talking about names, right? Names of plants and fungi?
Correct.
Okay, so they really wouldn't be the same. There would be two different sources. Um, probably the easiest to use for plants is the plant list, which is a joint, it's maintained by the Royal Botanic Gardens Kew, but a number of institutions contributed to it. It's a very easy user interface. Just very recently, within the past month, a new updated list has come out. It's called the Leipzig list, which I think if you search for Leipzig plant names list, you'll probably find it somewhere. But it doesn't quite have the nice user interface yet anyway. For fungi, the best source is Index Fungorum, which is, again, you'll find it if you search, is maintained by the by British mycologists, by the I can't remember the acronym now, but anyway, it's next door to Kew, right there, Commonwealth Mycological Institute. So those are the ones that we have used, and in both cases, they've been very generous to give us downloads of data that we can use in our own database. And thanks for joining from Nepal, by the way.
We have another great question, which I think is fantastic, and I'd love to hear your answer. How do you think is the best way in which herbariums create or promote the conservation of biodiversity?
Well, the basic answer, which is not the full answer, but the basic answer is that you can't conserve things if you don't know what they are. And the herbarium is the primary tool for plant identification. So fundamentally, we have to speak in a common language. We have to have a common name for organisms. And in case of plants, the place to get that is from a herbarium. You can use a book, but you'll often find that you're not entirely sure whether the plant you've identified is the right one. So you'll need to consult a herbarium. But beyond that, herbaria are a record of what used to grow where. We've been amassing these data now over 6 centuries. So we can get a sense of where plants used to grow, and we can compare that to where they grow now. And one of the more interesting tools that's been developed in recent years is using a GIS program and species modeling to be able to try to predict. If you know where species grew in the past and you can plot them on a map and see what the conditions are in those places, then you can make modifications to those conditions, sea level rise or higher rainfall or lower rainfall. And you can make predictions as to how species distributions will change in the future. We also use distributions of herbarium specimens to determine the extent of occurrence of a species and the area of occupancy. And both of these are key measurements or key metrics that are used by the International Union for the Conservation of Science for determining whether or not a species is endangered. So herbaria are extremely important for conservation.
So important to hear. Thank you so much, Barbara. I know your time is important, and so we won't keep you any longer, even though we have so many great questions that we could go on and on about. But thank you, Barbara, so much for taking the time to speak to us today.
It was my pleasure. Thanks very much.
Great. There's so much more on this topic to discover. So for the folks who'd like to learn all about it, you can purchase a copy of Barbara's new book yourself, Herbarium, The Quest to Preserve and Classify the World's Plants, from the NYBG shop. And we'll put a link for that in the chat at the close. So thank you everyone once again in our audience for joining today's presentation. If you missed any part of the event or wish to watch it again, a recording will be saved and shared online on the NYBG Lecture Library. We hope you'll join us for future science, conservation, and humanities lectures from the New York Botanical Garden. Upcoming in January, we're hosting 3 exciting plant science talks by newly emeritus curators, Dr. Andrew Henderson, Dr. Robin Moran, and Dr. Waite Thomas. Stay tuned for these invitations to learn more about plants and celebrate the careers of world-renowned scientists at NYBG. And for those who haven't already registered for our mailing list, go ahead and sign up using the link I will share here at the end. So until next time, stay well, stay green, and take care. Goodbye, everyone, and thank you once again, Barbara.